All right, in this video, we're going to compare the electron transport chain in the mitochondria to the electron transport chain in the chloroplasts. Now remember, plants also have mitochondria, and the mitochondria of plants do the exact same thing that the mitochondria of animal cells do, which is produce ATP that is necessary for all the various work that those cells have to do. Okay, so with that said, let's take a look at the mitochondria again. Let's remind ourselves that the mitochondria are made up of two membranes, the outer membrane and the inner membrane, which means there's two important spaces. There is the matrix, which is the space within the inner membrane, and there is the intermembrane space, which is the space between the inner membrane and the outer membrane. This figure you want to become very familiar with, because if you understand it completely, you'll really be able to understand the concept of the electron transport chain and oxidative phosphorylation, which involves the electron transport chain here and chemiosmosis, which is the production of ATP through ATP synthase here. So let's focus on the electron transport chain. Let's get our bearings. This is the intermembrane space out here, and this is the matrix down here. And you should identify the reactants of the electron transport chain. And I have arranged them for you in decreasing electronegativity, meaning that oxygen here has the highest affinity to electrons and NAD plus has the lowest affinity to electrons. And I showed them as what's called redox pair. This is oxygen before it grabs those electrons. This is oxygen after it grabs those electrons and hydrogens. So oxygen and water are a redox pair. Cytochrome C, when it's oxidized, when it's not carrying those electrons, is cytochrome C oxidized, and when it's reduced, it's cytochrome C reduced, and so on and so forth. So let's identify each one of these reactants within the electron transport chain. And when I say reactants, they are like any other products and reactants and intermediates, like any other biochemical pathway we've seen before, like glycolysis, like the citric acid cycle. Okay, so let's identify oxygen, water right over here. There's oxygen, water. Let's identify cytochrome C right here. Now, these figures don't show you cytochrome C when it's accepted the electrons and when it dumps the electrons off. So you're not going to see the oxidized, the reduced version. But know that it does get oxidized, it does get reduced. Let's look for Q. There's Q. And again, this figure won't show you Q and QH2, but know that Q is an intermediate, does get oxidized, does get reduced. And then there is FAD. Here it is right here. So there's FAD when it's oxidized, there's FADH2 when it's reduced. And NAD plus, NADH. Here it is, NAD plus, NADH. Now each one of these, right, these are intermediates of various reactions, meaning there are enzymes that catalyze whatever reaction that they are involved in, that catalyze these redox reactions. So what are the enzymes that catalyze the oxidation and the reduction of each one of these intermediates? Well, we have complex 1. That's right here. That's an enzyme embedded within the inner membrane of the mitochondria. We have complex 2. That's right here. We have complex 3. That's right here. And we have complex 4. That's right there. Okay, now what we want to do is we want to look at each one of the reactions that's catalyzed by each one of these enzymes and see, is it an exergonic reaction? Well, how do we do that? Well, reaction, when we're talking about a redox reaction, a reaction will be exergonic when the molecule that's being reduced has a higher electronegativity than the molecule being oxidized. Basically, what that means is as long as the molecule that's trying to steal electrons has a higher affinity for them, that is an exergonic reaction. So let's take a look at each one of these. So let's start with complex 1. Enzyme 1, complex 1. What reaction is it catalyzing? It's catalyzing the transfer of electrons from NADH to Q. Well, let's see. Is Q more electronegative than NADH? Here's Q way up here. Here's NADH way down here. Yes. So that's naturally an exergonic reaction, which means that that reaction is going to release energy. And that energy is coupled to an endergonic process. What's that endergonic process? Moving protons against their concentration gradient. And that's exactly what's happening there at complex 1. All right. At complex 2, let's take a look. FADH2 is being oxidized, which means Q is being reduced. Q is stealing electrons from FADH2. Is that an exergonic process? Is Q more electronegative than FADH2? Let's take a look. There's Q, high. There's FAD, low. So yep, that is an exergonic reaction. Now, that does release energy, but complex 2, because this is going to make it tricky for you folks, complex 2 doesn't couple that release of energy to the pumping of protons. Okay. However, it does transfer those electrons to Q. So we've got two different ways that we have reduced Q. We've reduced some of the Q via the NADH here in complex 1, and we've reduced some of the Q over here at complex 2 from FADH2. Okay, but either way, we've got a whole bunch of Q floating inside the inner membrane, right? There's not just one Q, there's lots and lots of Qs, and they're going to be involved in the next reaction, the reaction that occurs at complex 3. Okay, so complex 3 is going to catalyze now the transfer of electrons from Q to cytochrome C. Let's see. The only way that's going to be exergonic is if cytochrome C is more electronegative than Q. Is it? Cytochrome C at 2 here and Q at 3. Yes, that's an exergonic reaction. And so our complex 3, our enzyme, will couple that to the endergonic process of pumping a proton against its concentration gradient into the intermembrane space from the matrix. Finally, we have our complex 4. We have enzyme complex 4 that catalyzes, let's take a look, the transfer of electrons from our reduced cytochrome C to oxygen. Is that exergonic reaction? Well, let's take a look at the electronegativities. Yep, sure enough, oxygen is more electronegative than cytochrome C, which means that the reaction catalyzed by complex 4 is indeed an exergonic reaction, which means it can couple the released energy to the endergonic process of pumping protons against their concentration gradient. Fantastic. By the end of this, we now have our proton motive force in the intermembrane space. That source of energy, that proton gradient, is used by ATP synthase. As those protons move down their concentration gradient, ATP synthase uses that exergonic process and couples it to the endergonic process of making ATP from ADP and inorganic phosphate. That's called chemiosmosis, and that's how mitochondria produce the ATP that are then going to be distributed throughout the cell to do work. Let's compare the process that we've just discussed to chloroplasts. So, since in mitochondria here, we had to identify the structure of mitochondria, identify the important membrane and the important spaces, we've got to do the same thing with photosynthesis. Photosynthesis occurs in chloroplasts, and if you take a close look at chloroplasts, we have an outer membrane, we have an inner membrane, and then we have these thylakoids inside. So that's 3 membranes. So that means we've got 1, 2, 3 spaces. We've got the intermembrane space between the outer and inner membrane. We've got the stroma that is within the inner membrane, and then these thylakoid discs here, inside them, that's called the thylakoid space or the thylakoid lumen. Now, what is that important membrane in which those enzymes are bound? It's the thylakoid membrane. And so the two spaces that are going to become important here are the stroma outside of the thylakoid and then the thylakoid space in the thylakoid. So here we have it. Here we have the thylakoid membrane. Inside is the thylakoid space, and outside is the stroma. Now, the chloroplasts have almost the same purpose here, or at least I should say the electron transport chain of chloroplasts sort of serve the same purpose as mitochondria in that they make ATP. Unlike the chloroplast, that ATP that, unlike the mitochondria, the ATP that is produced within the chloroplast stays in the chloroplast and is used solely for the Calvin cycle, for the production of glucose. Now, in addition, in chloroplasts, you also get the production, again in the stroma, of an important electron carrier called NADPH. That's going to be important in the Calvin cycle as well, because remember, what we're doing in the Calvin cycle is we're taking CO2, that's extremely oxidized carbon, and we are reducing it into a carbohydrate. And when you reduce a carbon, you have to add electrons to it, and you have to add hydrogens to it. Where is that coming from? Right there, the NADPH. In addition, when you reduce carbon dioxide into a carbohydrate, it takes energy. Where does that energy come from? Right there, that ATP. Okay, so let's take a look at how this ATP, how this NADPH is made. Let's take a look at the electron transport chain that exists within chloroplasts. Let's identify it, and let's go through this the same way we did with the mitochondria. We first have to identify the reactants of the electron transport chain. Well, we have our old friend oxygen and water. Here it is, right there, oxygen and water. We've got plastocyanin, and the oxidized version, and plastocyanin, the reduced version. Where is that? Right here. Now, folks, plastocyanin is evolutionarily related to cytochrome C. And then we have plastoquinone, oxidized, plastoquinone reduced form. Where is that? Right there. Plastoquinone is evolutionarily related to Q that we saw in the mitochondria. And like Q, PQ exists here within, it's hydrophobic, so it lives within the membrane. Plastocyanin, since it's hydrophilic, it exists out here, floating around, associated in the outside of the membrane. Then we have, let's see, we've got our NADP plus NADPH. That is right here, NADPH, NAD plus. That is similar in structure to NAD plus and NADH. And finally, we have ferredoxin. FD plus is the oxidized form, and FD is the reduced form, and there is our ferredoxin. So now we have identified our reactants, the molecules that are going to be reduced and oxidized as electrons are transferred from one molecule to another. Now we have to identify the enzymes. What are the enzymes that are going to catalyze these transfer of electrons, these redox reactions? Well, first we have photosystem 2, that's right here. Then we have the cytochrome complex, not to be confused with cytochrome C. This is the cytochrome complex right there. And then we have photosystem 1 right there. And then we have NAD plus reductase. And that is that should be NADP plus reductase. And that is right there. So we have all of our reactants. We've identified all of our enzymes. Let's approach this like we approached the mitochondria, the electron transport chain there. In that, a reaction is going to be exergonic if the molecule stealing the electrons is more electronegative than the molecule who's having the electrons stolen. So let's take a look here at photosystem 2. What is being reduced and what is being oxidized? Well, we're going from water, the reduced state of oxygen, to oxygen. So water is being oxidized. It is losing electrons, and where are those electrons going? To PQ. So let's take a look. Is water less electronegative than PQ? Or let's reverse that question. Is PQ more electronegative than water? Well, here's water oxygen up here. Here's PQ way down here. Whoa, wait a minute. That is not an exergonic reaction. That's an endergonic reaction. We're trying to use something that doesn't hold on to electrons very well to steal electrons from water, which was in animal cells like the single most electronegative molecule. So that, in a nutshell, folks, is the problem that chloroplasts have evolved to solve. How do plants steal electrons from water? Well, the answer is in the enzyme itself is a special chlorophyll called P680. And this chlorophyll is a pigment molecule that absorbs very specific wavelengths of light. In fact, 680 nanometers wavelengths of light. Now here's the neat thing. P680 is inherently even more electronegative than oxygen. So P680 would be way up here, way above oxygen, way above water. Which means P680 spontaneously, no problem, steals those electrons from water easy. However, we still got a problem. How is PQ going to steal those electrons from P680 if P680 is even more electronegative than water? It can't. That's where light comes into play. When P680 absorbs sunlight, what happens is, first of all, it absorbs, here is a chlorophyll right here, it absorbs right around between 400 and 500, and again between 600 and 700. Notice it doesn't absorb green light. Right? These chlorophylls look green because they reflect green. They don't absorb it. Now, when they absorb these wavelengths of light, what happens? Well, here we've got P680. It has spontaneously stole electrons from water, and now it's just holding on to them. But then it absorbs a wavelength. It absorbs light, and its energy of electron goes way up. It becomes much less electronegative. Aha. So when P680 absorbs that light, it becomes way less electronegative, so much so that PQ now is suddenly more electronegative than it, and now PQ can steal electrons from P680, no problem. All right, next we have cytochrome complex. That's our next enzyme, and it catalyzes the transfer of electrons from PQ to PC. Let's take a look. Is this an exergonic reaction? Let's find out. Is PC more electronegative than PQ? PC is up here at 2, PQ is over here at 3. Yep, absolutely. Which means this is a naturally exergonic reaction. We don't need sunlight to do any funny business here. And in fact, this reaction, since it's exergonic, releases energy, and that energy is used to pump protons from the thylakoid membrane into the thylakoid space. Okay, let's take a look at the next. So we're at our photosystem 1. This is our next enzyme. And it's catalyzing the transfer of electrons, so the oxidation of PC and the reduction of FD. Is this reaction exergonic? Is FD more electronegative than PC? Let's take a look. Here's PC way up here, but whoa, here's FD all the way down here. This is an endergonic reaction. So let's take a closer look at photosystem 1. Deep inside there, we have a pigment molecule called P700. And P700 is just like P680 in that it is more electronegative than PC. So it has no problem stealing those electrons from PC. So there's those electrons from plastocyanin sitting in photosystem 1 and being held very tightly because, again, the electronegativity of P700 is way higher than that of plastocyanin, but that means it's also higher than FD. So we got a problem. How is FD going to get those electrons? Well, our special pigment molecule is going to absorb wavelengths of light. That's going to excite its electrons, which makes it less electronegative. It can't hold on to those electrons very well. Awesome. Now FD can grab those electrons. So now that's how FD becomes reduced. All right, let's look at our last enzyme, our NADP plus reductase. And what that is catalyzing is a transfer of electrons from FAD to NAD plus. Okay, so basically the oxidation of FD and the reduction of NAD plus to NADPH. Let's take a look if this is an exergonic reaction. Is NAD plus more electronegative? Yep, sure is, which means this reaction is exergonic. And that also means we don't need any funny business here. We don't need any light. It's a naturally exergonic reaction. However, unlike the cytochrome complex here, it does not then couple that to the pumping of protons into the thylakoid space. All right, so there is our final electron acceptor in chloroplasts is NADPH. The electron donor in chloroplasts is water. Right? That's completely reversed than what we saw in mitochondria. In mitochondria, the initial electron donor is NADH, and the final electron acceptor is oxygen and water. In mitochondria, they're opposites of each other. And now we have this proton gradient here in the thylakoid space. And it then is used as it goes down its concentration gradient through our old friend ATP synthase, which couples this exergonic process of proton going down their concentration gradient to the endergonic process of making ATP from ADP, an inorganic phosphate. And there we have it. Now again, in all cells, animal cells and plant cells, the ATP that are going to be used cellularly wide is made in the mitochondria. In the chloroplasts, the ATP that's made, the NADPH that's made, is going to go into the Kelvin cycle, where CO2 is going to use the energy of ATP and the electrons and hydrogens of the NADPH to make sugar. And that's the job of the Kelvin cycle, and we'll get back to that in another lecture.